Welcome back. The next type of gene transfer that we're going to cover is the process of transduction. Now, with conjugation, you had a physical structure that existed between the two bacteria. With transduction, this is going to use a bacterial virus to transfer the genetic information. And we haven't covered viruses yet in class. So I'm going to kind of give you a brief outline of what the bacterial virus is that is going to do this transfer first. And that virus is called a bacteria phage. Sometimes you will hear it referred to just as a phage. It's called a T4 bacteria phage, and that T4 just gets to the overall structure of the bacteria phage. So what you're going to have, I'm going to kind of draw this structure here. You're going to have a capsid, okay? That capsid is going to be made of protein. And what's going to happen inside that capsid, this is where you've got the viral DNA. So that DNA is housed inside the head of the virus, because you'll sometimes see the capsid referred to as a head. All right? And with the phage, then what it's going to have, I'll draw this a little thicker here. Okay? It's going to have a tail. And what's neat about the tail is this part of the bacteriophage tail has the ability to contract. And that's going to be important because what this bacteriophage is ultimately going to do is it's going to inject, kind of like a hypodermic needle, that viral DNA into the bacteria. Okay? So this is going to be, the tail essentially is how that viral DNA will leave the virus particle. Okay? And then the neat thing about this, because I think bacteriophage kind of look like they are something that landed maybe on the moon, you've got the tail fibers. And the tail fibers then are going to be what are going to sit down on the bacteria and make contact with the bacteria in order to inject that DNA into the bacteria itself. Okay? So with bacteriophage, it's going to be composed of DNA, so it's going to have some genetic material, and it's going to have protein associated with it. Okay? The protein then is the capsid and the tail that's involved in housing the DNA and also transferring then that DNA into the bacteria. Okay? So what can happen with the bacteriophage? Okay? There are two types of infections that you can have with transduction. You can have generalized transduction that happens. And that generalized transduction is kind of a random assortment of genetic material that would be transferred to the bacteria. You can then also have specialized transduction. And really the specialized transduction, we're talking about usually one specific gene that is transferred. And this is the way that scientists can use bacteriophage, again, to do something that we want it to do. So specialized transduction usually involves a lot more genetic engineering on that end. Generalized transduction will happen out in the environment, and I'll talk about how that bacteria, or how that bacteriophage picks up additional DNA. Okay? So we'll talk about generalized transduction first. Okay? With that generalized transduction, what happens? And obviously my bacteria here is not to scale. This is going to be our unexpecting or unassuming bacteria that's just hanging out in the environment, doing its thing. And then what we have happen, okay, you're going to have the bacteriophage that will sit down onto the bacteria. And what it's going to do is it's going to inject its DNA, because I should here, this is our bacterial chromosome. We've also got our viral DNA here. So what happens, that contractile sheath will sit down, and what happens is that DNA is going to be injected into the cytoplasm. And then what happens, because that bacteria has ribosomes, because it undergoes the process of transcription and translation, that bacteria is going to make copies of the viral DNA. And what will end up happening for this poor, unassuming, unexpecting bacterial cell is now, instead of making the proteins that the bacteria needs, that bacteria now is just going to pump out all kinds of viral particles. And I'm only going to draw a few in here, but essentially what would happen is you're going to have the bacteria become filled with viral particles. So you're going to get massive production of viral particles. And inside those capsids will be viral DNA. I'm going to draw two of them here with just the pink representing the viral DNA. But you can also then have some of the bacterial chromosome that is picked up in the viral capsid. So in addition to the DNA that makes up the virus being packaged in, sometimes, because especially when this gets later and later into production, that bacterial, the inside of the bacterial cell starts breaking down, the genome, the DNA starts to be broken apart, and bits of the DNA can be captured inside of the bacterial phage capsule, all right? And then what happens, the way that those particles are released is going to be by cell lysis. So once that bacteria phage sits down on that unassuming bacteria, that bacteria's lifespan has now become limited, because it's going to pump out all kinds of viral particles until that bacterial cell bursts open and releases the viral particles into the environment. That now allows those bacteria phage to infect other nearby bacteria. Some of them may just contain the viral DNA. Some of them may contain bits of the DNA from the bacteria that it infected before. And that can then be transferred to the new bacteria, okay? Part of this that I haven't told you about yet, because we haven't talked about viral replication. With this process, there is what's known as a lytic phase, and that's what I described here. You end up with bacterial cell lysis. And then there's also the lysogenic phase. And in that case, the easy way for me to describe this right now, not having talked about the viruses yet, is that that virus essentially is going to be dormant. So instead of going in and pumping out all kinds of viral particles, it kind of hangs out there in the bacterial cell. And if it's one of the ones that has the bit of extra genetic information, that genetic information can be passed on to the bacteria that picked up that bacteria phage, okay? So that's kind of a brief overview of the process of transduction. The next video, I'll go over the process of transformation, okay? If you have any questions, please let me know.